All right, so let's talk a little bit now about the structure of the transmembrane region of a protein. So what I first want you to consider is what's the environment inside the phospholipid bilayer. So hopefully by now you realize that the inside of that phospholipid bilayer is hydrophobic. That's where those fatty acid tails are congregating together to get away from water. All right, so we have the polar head groups out here. Right, so the polar head groups, and then we have the fatty acid tails. That interior part of the phospholipid bilayer is hydrophobic. So, what type of side chains would be best to interact with the transmembrane domain? Well, we have our nonpolar side chains. And those nonpolar side chains are going to interact very well with the hydrophobic interior. All right, but there's something else to consider. All right, so this makes sense. The nonpolar amino acids are most likely going to be the ones that are crossing the transmembrane domain because those are the ones that are happy interacting with the hydrophobic phospholipid bilayer inside this kind of transmembrane region. But we still have a bit of an issue because remember, we have the backbone part. All right, so our R group can be nonpolar. That works just fine. But if we still have the amine, we have the alpha carbon, and we have the carbonyl group, and this is attached to amine, alpha carbon, and carbonyl. And so basically, no matter what, these are still polar. That's a polar bond, nitrogen, hydrogen. This is a polar bond, carbon, oxygen. And so even if we have our nonpolar amino acids crossing the membrane, we still have the backbone to deal with. And so this is one of the reasons that most transmembrane proteins are going to form an alpha helix. And it also is the same reason that bacteria use beta-pleated sheets. So basically, the secondary structure of alpha helices and beta-pleated sheets allow the polar backbone to be hidden from the hydrophobic membrane. So what do I mean by that? If we take a look at our alpha helix here, we see that our alpha helix basically spirals around, as we know, but goes straight up and down. And what's key here is each amine group is hydrogen bonded to each carbonyl group. And this is actually 4 amino acids away. All right, so here we have the carbonyl, central carbon, amine, carbonyl, central carbon, amine, carbonyl, central carbon, amine. So this carbonyl here is 1, 2, 3, 4 away. And so basically in doing so, all of these hydrogen bonds are internalized straight up and down. All right, all these hydrogen bonds are internalized straight up and down. And we saw this with the when we looked at the aquaporins, because those hydrogen bonds being straight up and down hide these polar residues from the transmembrane phospholipid tails that would be all around it. Another thing is that the transmembrane region of an alpha helix is about 20 amino acid in length, right? But that might vary. Why might it vary? Well, remember, we actually have lipid rafts. So we have regions of the membrane that actually might stick out a little bit further in our lipid rafts and so forth. So there are variations here. And this is actually one of the reasons that the lipid rafts can be used to aggregate transmembrane proteins together. So if I have a transmembrane protein who has an extra long hydrophobic region, that extra long hydrophobic region is happier here than it would be here, because now we still have hydrophobic regions sticking out. So that's why those lipid rafts with their longer fatty acid tails and therefore their wider membrane portions that look like islands floating around, that's why they can be used to basically isolate or localize transmembrane proteins that have longer regions. And so if I have transmembrane protein A and transmembrane protein B, and both of them have these extra long region, they're both likely to be inside that lipid raft, and that's like being stuck on a deserted island together. They're much more likely to find each other and create that quaternary structure, right? So that's why that transmembrane length may vary a little bit, but generally they're around 20 amino acids. And here's what the alpha helix does to the side chains. Every one of these side chains ends up sticking outward. All right, so even these over here, these are going to stick outward kind of behind. They're going to go behind there. These are going to come in front. So they're always sticking outward. So this is looking straight down at our alpha helix here, straight down. And you can see the carbonyls and the amine groups, amine in blue, carbonyl in red, they're straight up and down, but the side chains are all sticking out. So basically we have these side chains pointing out all around as it goes. And that allows those side chains, which are mostly going to be nonpolar, to interact with the phospholipid bilayer all around it. So let's think about this a little bit further. This means that we can use tools called a hydropathy plot to really try and identify where transmembrane regions might be. Because if we're using alpha helices, there's going to be a long stretch of about 20 amino acids that are going to be the majority of them are going to be nonpolar. So what does a hydropathy plot do? A hydropathy plot, in its most simple way of thinking about it, measures hydrophobicity. So the higher you are on the hydropathy plot, the more hydrophobic. How does it do that? Well, basically it's measuring the energy it would take to dissolve that in water. So if you're hydrophobic, it's going to take more energy to dissolve that in water. And if it's polar, a polar region, it's going to take very little energy to dissolve it in water. And so if you have stretches where you have nonpolar side chains intermixed with polar side chains, they're probably going to even out and at the very least be around here, or maybe even fairly simple to dissolve. But if you have long stretches of 20 amino acids where the majority is nonpolar, that's going to be very hard to dissolve in water. And so your hydropathy index is going to be high. This is probably a transmembrane region because it's a region with a long stretch of hydrophobicity. So if we look over here in the bacterial rhodopsin, how many transmembrane domains does this have? Well, we can look at the hydropathy plot and it probably has 1, 2, 3, 4, 5, 6, 7. It has 7 regions with pretty high hydropathy. And so that's 7 nonpolar regions. It's probably a 7-pass transmembrane domain with alpha helices. Now, what does that mean? Well, that means we have our membrane here, all right? And so I'm not going to draw the phospholipids, but that's what this would be. That means that there's going to be 1 pass, 2 pass, 3 pass, 4 pass, 5 pass, 6 pass, 7 pass. And they have to be connected, all right? So there's your first transmembrane domain, second, third, fourth, fifth, sixth, and 7th. But we can find those transmembrane regions using our hydropathy plot because we know that they are going to have much higher levels of nonpolar amino acids in that stretch of 20. Now, we also talked briefly about beta barrels. So now these are mostly prokaryotic organisms, as well as the outer membrane of mitochondria and chloroplasts, which we know have their evolutionary origin from prokaryotic organisms. Why these? Well, same thing. The carbonyl and the amine group are going to form hydrogen bonds, and they're internalized within these beta sheets here. So this also hides the backbone, the polar backbone, from the hydrophobic region because all of those interactions would basically be kind of internalized in our beta sheets here. Now, with this, the R groups opposite point out. So the first one's here, the next one's pointing the opposite direction. Whereas in our alpha helix, remember they were all pointed outward kind of around our spiral staircase. These are pointed outward and inward in opposite directions. And so if you think about it, if we have this transmembrane protein, it's going to make sense that every other one is going to be pointed outward and probably nonpolar. But what do you think the ones pointed in would be? Well, the ones pointed in are probably going to be polar because we're making that polar channel for a polar molecule to get in. So if this is outward into the membrane, and then over here we have inner channel, all right, so this is the inside of the channel, These are probably going to be nonpolar side chains, and these are going to be polar, because the ones facing outward have to interact with the hydrophobic membrane. The ones facing inward are going to interact with whatever polar molecule is being transported. So, question, would you be able to predict beta barrel proteins from a hydrophobicity plot? So from those hydropathy plots, that's aka hydropathy plot, would you be able to predict a beta barrel? Well, the answer is no, because it works with an alpha helix, because the vast majority of those side chains do interact with the membrane and they're going to be nonpolar. Whereas here you've got every other one, so only 50-50 are nonpolar, so they're not really going to have that high level of hydropathy beyond anything else. So beta barrels are not predictable usually by hydropathy plots. Alpha helix transmembrane regions definitely are. All right, last thing. Transmembrane proteins are very difficult to study, and this is because they have those long hydrophobic regions. And so if you just try and dissolve that, that's not going to dissolve in water. It's very happy in with the membrane. You can even break apart chunks of the membrane, which actually itself is hard to do, but that membrane protein is not going to become isolated by itself. All of these things are going to crash out of solution because those long nonpolar regions cannot dissolve in water. If they could, then there would be no reason for this transmembrane domain basically to lodge itself in that hydrophobic region. So how do we isolate membrane proteins and study them? Well, this is where we use detergents, and we've seen detergents way back with our bubbles. Our detergents have a polar head and a single tail, and so they're going to create these micelles that are spheres, kind of like this. If they have two tails, like a phospholipid, then they create a bilayer. If they have one tail, they create these little micelles. But these micelles have polar heads, and then they have the nonpolar interior. The nonpolar interior here. And so detergents are happy to break apart the membrane because they can interact with the membrane components the same. And they can also interact with the transmembrane domain, because now this membrane protein can basically work itself in through this micelle here just the same, because that nonpolar region here is very happy on the interior of the micelle. And we see that happening right here. So detergents can be used to isolate membrane proteins because the detergents have a nonpolar region and the polar head groups. So they help dissolve that nonpolar region of the protein into water by basically kind of incorporating it into its micelle.